Hello and welcome to my video presentation. We have a lot of fun stuff in store for you today. We will be exploring yeast and bacteria up close and personal as I conduct two separate experiments on them. Yeast are a type of bacteria and fungi. Scientists disagree about this, but one thing is certain that they ferment, meaning that produce alcohol and carbon dioxide. The bacteria being used are photosynthetic, meaning that they can absorb carbon dioxide by creating their own food in the form of glucose, which feeds the yeast. I used several different types of materials. I used 5 vials of Protists. I used one of Procentrum, Amphidinium, Gymnodinium, Cyclotella, and Chlorella. I used rubber bands, and I used balloons and 50 ml graduated cylinders, and I also used 2 types of yeast. I used Heishmann's Quick Rise, and I also used Red Star Instant Yeast. I also used the pipettes and test tube rack from my local biology classroom. What I did first was that I made the yeast become active live yeast. So what I did was I heated water to 110 degrees Fahrenheit. I put it into a beaker and I let it cool. I had it at 120 and let it cool down a little bit to 110. And then I distributed it evenly among my graduated cylinders. I put 28 mLs into each, and then I put 1 teaspoon of each protist, 1 teaspoon of glucose, and 1 tablespoon of yeast into the solution. I let it sit for 30 minutes after the bubbling reaction occurred, which proved the live yeast were alive. And then I recorded my data, and I repeated this process 3 times to get 3 separate tests that were identical to each other with very little variation in between. That means that they follow the scientific method. And the reason I chose these 5 types was that I used a mixture of algae, diatoms, and just some other very commonly known types of Navicula, which is a subgenus of the kingdom Protista. So those were the ones I chose. I chose 2 diatoms, 2 algae, and 1 just Navicula subspecies. The importance of glucose, when I met judges actually, they were wondering why you use glucose in this, and the importance of glucose is that without having some energy source, the yeast cannot break down the fecal matter of the protist, which then produce the plant oils. So they need to have some energy source which comes in the form of glucose. That answers that question. My data showed that Amphidinium and Gymnodinium were the best, although Procentrum might be a possible solution, but through my research, Amphidinium or Gymnodinium would be the best. If I were to apply this to the real world, I could say that if we were to have large national laboratories or universities having large aquarium-sized places where we could hold all these protists and add yeast, so it would be a self-sustaining environment, we could actually raise these and get plant oils and extract them and refine them, and potentially make a long-term, more energy efficient, less harmful alternative to petroleum, and it would make less carbon emissions, and it would also be easier in extracting it than it is from pumping oil out of the earth. So this is just one application. You could also use it to get biometric data and different things for maybe trying to using these on something like a cure for cancer, Alzheimer's, or some other disease where they might be useful. It would just take further tests and research to find out. I would like to say that my hypothesis was a failure, because I did get my purpose was a success. I fulfilled my purpose. I was supposed to see which protist was the most efficient, but my hypothesis was incorrect. I said that Chlorella would be the most effective, but in actuality, Amphidinium and Gymnodinium were the best. So that would explain why my hypothesis was incorrect. Thank you for watching my video presentation. I hope this was very informative, and I hope that this opens up your knowledge about protist and the interaction with yeast and plant oils and petroleum alternatives, and maybe using something that isn't so harmful for the environment and that emits less carbon dioxide. Have a nice day.